Welcome and good evening. I am Dr. Carl Longley of Fresno State's California Water Institute, and on behalf of Fresno State and Dr. David Soldowski, the director of the California Water Institute, it's my pleasure to welcome you here this evening to listen to the very excellent panel to whom I will introduce you to. We have Dr. Brian Dennis, Dr. Carol Lee, Dr. Joseph Thorley, Dr. Walter Pearson, and as a moderator for the panel session, Dr. Terrace Quinn. Dr. Terrace Quinn, and the moderator is Dr. Robert Zink. Excuse me for my files up there. California Water Institute got underway about 11 years ago under with the help, I might add, of then California Senator Jim Costa, who's here this evening with us. And the California Water Institute is involved in a number of important studies. They include water use efficiency, drinking water and wastewater services for disadvantaged communities in California, and the implementation of the integrated water management planning. Water is so critical to California's environment, its economic health, and its future. Over the last 6 decades, the water landscape of California has changed drastically with the building of dams, reservoirs, and conveyance facilities to protect Californians and their property from flood damage, to provide water for drinking water and growing the crops that we consume, and to generate energy. An individual who has been an important part of the California's water history and will continue to be a very important individual shaping California's water policy well into the future is the now Congressman Jim Costa of California's 20th Congressional District. It is my great pleasure and honor to welcome Jim to the podium this evening.
Thank you very much, Dr. Longley, for those kind words. Carl and I, along with David Soldowski and the California Water Institute, working with the International Water Institute here at Fresno State, have, I think, done a terrific job in providing an opportunity for us to focus on the important policy issues affecting water use in California. Not only in terms of its historical use, but what we are doing today to ensure that in the 21st century, Californians will have a stable, secure supply of water that will be able to address the issues of the environment, and at the same time ensure that for future generations of Californians, we will have the opportunity to continue the economic advantages that California has had as a state really throughout the 20th century. But those advantages today come into question, and they come into question for a number of reasons. We have a water system that was designed for approximately 20 million people. Today we have 38 million people living in California. By the year 2030, it's estimated that we will have 50 million people living in California. And so the demands on our water resources continue to be great. And this evening's workshop is a second workshop that we've initiated as we deal with the difficult challenges of the balances of ensuring a long-term water supply, a reliable water supply, focusing on efforts on restoration of the environment, and at the same time dealing with the population growth that is going to, just as much as our agricultural communities, require a stable source of water throughout the 21st century. And so the workshop here today is really to look at how we balance those issues. The focus today, of course, is the biological opinion, primarily focusing on Delta Smelt and its interrelationship to the other elements of the environment within the Delta. But the workshop today from our esteemed group of scientists who we're honored to have here at our university goes beyond that, because part of the dilemma or the challenge we've had in California among the 6 regions of the state is trying to allocate shortages. And clearly, when you're trying to allocate shortages of a water supply, you have not only the tension, but you have the fights. Those are not new. I think we all know of hearken of the Mark Twain observation when he was in the West, that it was clear that in the West, whiskey was made for drinking and water was made for fighting. And we clearly have a history of decades on fighting over our water resources. But we have an effort that is ongoing here with the Bay Delta Conservation Plan that we hope will allow us a path to begin to fix some of the water challenges through thoughtful solutions that will ensure that every region of California has a stable supply of water well into the 21st century. And the way we do that, of course, is by first working together, secondly, by using all the water tools in our water management toolbox, and third, ensuring that as we're talking about efforts on balancing the needs of water supply and water availability, knowing that we have, as this last winter, a abundant rainfall and snow in the Sierra, and as we had the previous 2 winters below average snow and rain in the state. That is the cycle that we know will continue to exist in California in the future. We have about 150 years of recorded climate in this state, but that is just a small snapshot when you look at the entirety of climate history that has occurred for millions of years. And we know that the climate is changing. And clearly, we need to focus on how those changes are going to impact our water supply and how they're going to impact the environment as the state continues to grow. So I am really looking forward to listening to the experts, the scientists here, as they talk about the underlying science as it relates to the biological opinion with Delta Smelt, its impact to the other fisheries that clearly are of concern with regards to salmonoid. We have a separate biological opinion on them. As we look at the other factors that involve temperature change, as it looks at various impacts of non-point source runoff that has impacted both the Sacramento and the San Joaquin River Delta system, as we look at other discharge issues that have impacted the Delta. All of these questions, as we try to quantify the ability as to whether or not the underlying science is as good as it can be. And of course, we know with recent court decisions that have taken place that much of that science that has been used to implement the recent biological opinions has been brought into question. And so we look forward to hearing our scientists that come from a number of well-respected universities throughout the country as they give us insight as to what is the best science available as we consider the challenges in fixing California's water system for the long term, for a growing state, for the future of California. So I've said enough. I'm looking forward to hearing our panelists. And Dr. Longley, Carl, who is the, I think I'm going to turn the program over for you because I don't actually have an agenda in front of me. So thank you very much, all of you. And I look forward to working with all of you.
Thank you, Jim. Next, we are honored to have the Honorable Craig Manson to describe to us the roadmap and describe the legal context of the discussion here this evening.
Thank you. For those of you who don't know me, I am Craig Manson. I'm the Executive Director of the Center for Environmental Science Advocacy and Regulation, formerly known as the Council on Endangered Species Act Reliability. I, over the last 20 years, have served as the General Counsel of the State Department of Fish and Game, as a Superior Court Judge in Sacramento, and as the Assistant Secretary of the Interior for Fish, Wildlife, and Parks. When I left that job in Washington, I returned to Sacramento as Professor of Law at McGeorge School of Law in Sacramento. It was during that time that I founded CESAR, the nonprofit that is one of the sponsors here today. It's a volunteer-driven organization, but its mission is to ensure that the best science is used in public policy and decisions. I'm a public policy expert, not a scientific expert, but I know that public policy decisions must be informed by the best available science. And my motto is, we go where the science leads us. And so we did this seminar like this about 2 years ago, and it gave us an insight into many, many issues that involve the Delta Smelt and the Delta. And over the last couple of years with the litigation and the controversy about the science, it's time to take another look at that science. And these distinguished individuals have done just that, and we're very proud to be able to present them to you and get their input. We at CESAR, as I said, don't do anything but go where the science leads us. So sometimes you'll see us supporting an endangered species listing. Sometimes you'll see us opposing it based upon our view of the science. We have a scientific advisory board that advises us in that regard. And as a matter of fact, just today the New York Times reported that the Fish and Wildlife Service has found that it may be warranted to list a species called the American eel along the East Coast. And that was as a result of the CESAR petition that they came to that decision. And so we're proud to play an important role in protecting endangered species and enforcing other environmental laws, but only where the best available science supports the action. And I want to thank the university and the California Water Institute. It's always good to see Congressman Costa, who I got to know about 20 years ago when we worked together and doing some things for California when he was in the legislature. And I appreciate his support as well. And I thank all of the scientists who have come today. And I had the chance to have breakfast with them this morning, and they had a lively discussion. And it's just fascinating. So thank you very much for coming, and I hope you get something useful out of this for yourselves. There will be an opportunity for you to interact with the panel as well. Thank you.
Thank you, Judge Manson. We are now going to introduce you to Dr. Robert Zink and turn the rest of the program over to Dr. Zink.
Thank you. I trust you all realize how humbling it is to follow a dean, a congressman, and a judge. Not an easy task. But what we're here this evening to do is to discuss, as you've heard, what the best science says about the situation in the Delta as regards the management of water and the preservation of Delta organisms, especially with regard to Delta Smelt. As Dr. Longley mentioned, I'm Robert Zink. I'm going to moderate this session. And first, I'd like each of the panelists to just, again, remind you of their names, tell you where they're from, and give you a very brief overview of their area of expertise, beginning with Dr. Lee.
Yeah. Can you hear me? My name is Carol Lee. I'm a professor at the University of Wisconsin-Madison, and my work is focused on physiological genetics and biogeography of zooplankton, the prey items that the Delta Smelt feed on, specifically on the species that they feed on, as well as a variety of invasive species, plankton, benthic invertebrates. And my work on biogeography has led us to sample a wide variety of estuaries, all the way from Alaska to Baja, Quebec to the Gulf of Mexico, as well as Europe and Asia. So I've looked at a lot of the estuaries throughout the world.
Hi there. My name is Joe Thorley, and I'm a registered professional biologist from British Columbia in Canada. And I specialize in fish population biology. I run my own consulting company, Fresno Consulting Limited, and the majority of the work involves quantifying and predicting the effects of flow manipulations on fish abundance.
Hi, I'm Terry Quinn from the University of Alaska. I've been at the University of Alaska in the School of Fisheries and Ocean Sciences for the last 25 years. They keep me on. And I've been involved in a number of studies for the National Sciences as well. I was one of the panel members back in 2009, and so I guess I didn't blow that either, so it's good to see me once again as well.
Good evening. My name is Walter Pearson. I've worked as a fisheries biologist and doing research for the Marine Sciences Lab in Spring, Washington for over 25 years. I've taken retirement about 6 years ago and kind of been a consultant since then. The bulk of that professional experience has been trying to figure out why fish do the things they do, of course, in terms of behavior and later in terms of populations, and also to figure out how you're going to separate impacts or declines, understand what the causes were, and whether the mankind's activities led to that cause, whether it's natural factors.
Hello, my name is Brian Dennis. I do research in applications of statistics and modeling in ecological problems and natural resource management. I have a joint appointment as professor at the University of Idaho in the Department of Fish and Wildlife and the Department of Statistics.
Great. Thank you very much, panelists. We appreciate your taking the time to be with us this evening. We've chosen these individuals for obviously several reasons. First, they all have impeccable scientific records. They've published on water quality, fisheries biology, populations. And second, we consider all of them to be Delta Smelt neutral. That is, they haven't studied the species formally, they haven't gotten funding to do so. And this is important to us because you might think that we would choose people who had spent their careers studying the Delta and its organisms. But we wanted to ask qualified people outside of the community here to comment on the science involved, because this way they're not forced to comment on their own work, that of their colleagues, that of their students. And as we all know, scientists are totally immune to criticism. But at least this way they're not left in a compromised position, and they can look at what is known and what needs to be known from a fresh perspective, unbiased by personal involvement. So this is the essence then of the scientific peer review process. It's what is expected by the National Academy of Sciences. Guidelines for independent peer review is required under the OMB Information Quality Act, and it was explicitly asked for by Congressman Costa. We sent each of the panelists a list of questions dealing with some, but not all, of the main issues around water quality and particularly Delta Smelt. We sent them a list of over 50 references, long, very complicated scientific technical papers, asked them to read as much of it as they could, and then bring with them their answers to these questions. Now, obviously, we're not going to produce a comprehensive solution to all of the issues that are involved. We are instead providing a scientific status report, if you will, that builds on the research that has been published subsequent to the biological opinion issued by the Fish and Wildlife Service on 28th of August in 2008, which is especially relevant in light of Judge Wenger's recent ruling on this biological opinion and the need for its modification. So we've attempted to provide these panelists then with information they'll need, or that one would use in considering what might go into the next biological opinion. So I'm going to read each question, hopefully they'll be clear, and then I'm going to ask the panelists to comment. We're not going to go in any particular order. Some of the panelists don't have expertise that deal with some of the questions, so not everybody will speak to every issue. Instead, they'll stick with the areas of their own expertise. And then, as was mentioned earlier, we're going to solicit written comments from the audience after we're done with this set of questions, and the panelists will then respond to them. So for the meantime, we ask you to hold on to your questions, write them down as they come to mind, so that we can address them after the first part of the program is completed. So our first question that we'd like our panelists to consider is this. Are the data before you consistent or inconsistent with the hypothesis that the variable X2 has a direct effect on the abundance of Delta Smelt? Would you like to take a stab at that?
Should somebody define it first?
Go ahead.
So X2, probably all familiar with it, in case you're not, denotes the distance in kilometers along the axis of the estuary from the Golden Gate Bridge to the 2 practical salinity unit. It's basically a measure of how saline the water is. So you go from the Golden Gate Bridge to where you get a certain salinity, kind of a low salinity, and that's it. So you can measure how much the fresh water is encroaching in the estuary. And so the question we were being asked was, does the location of this isohaline have a direct effect on the abundance of Delta Smelt? And my response to that was that multiple statistical analyses have failed to detect a clear relationship between X2 and Delta Smelt abundance. It is therefore unlikely that variable 2 has a direct effect on the abundance of Delta Smelt, although it may interact with other variables such as turbidity and copepod abundance and composition. So in layman's speak, what it's basically saying is X2 alone doesn't have a linear relationship. You can't say, well, if you have this value of X2, you're going to have this number of Delta Smelt. That's not to say it may not be important, because it may, depending on other variables, X2 may have different effects. So it's not just X2 alone. It may, whether X2 is important or not, may depend on the status of other variables in the estuary, such as turbidity.
Although I would like to add that based on the current measurements, we can't definitively say whether or not X2 is important. However, I would like to state that some of the potentially key variables, as they are measured, might not have been measured in the right way. For example, if you look at the food that the fish, the Delta Smelt feed on, the copepod, the zooplankton, their habitat is shifting quite dramatically as X2 shifts from, say, 72 to 94, for example. So from Susan Bay up to the mouth of the Sacramento River, that's a pretty dramatic shift in location of that low salinity zone. And that low salinity zone is where the estuarine turbidity max tends to be located. And that's where there's high levels of productivity of both the algae that the copepods feed on, that's the smelt food, and the productivity of the zooplankton as well as the smelt. So this is locating from a wide open bay to the mouth of a narrow river. And so what people have generally measured is zooplankton density, the numbers per cubic meter or the numbers per mil, rather than total census size or abundance or biomass. So I think people are measuring the wrong value when they're comparing these two habitats. So they're saying this change in habitat might not have a shift, might not have any effect. One of the key problems is how these particular things are measured, such as the amount of primary productivity, the amount of phytoplankton that's being produced, as well as the amount of zooplankton that's being produced. And that's something that sustains the fish. Did that make sense? Or was that too long-winded?
I'd like to address this in something of a historical context to give you a feeling about why X2 has been so important. Many of the studies that were done in the mid-2000s came up with the relationship between X2 and the abundance of Delta Smelt, the way it's measured in the trawlnets. They have surveys that date back into the 70s for. So there seemed to be this relationship between abundance and X2. However, when we had our 2009 panel, we concluded that the way that they had done those analyses was flawed. and that they were not looking at the right variables. Rather than looking at abundance, they should have been looking at the population dynamics that involves the mortality effect that is related to this particular variable. When the population dynamics are done correctly, then very few studies have found the relationship with X2. And so consequently, the scientific attitudes about that have shifted. The National Academy of Sciences published a study last year that said that conceptually there could certainly be a relationship between X2 and the dynamics of Delta Smelt, but it doesn't have to be that way. And so the empirical evidence right now basically says that X2 does not seem to have any weak association, or the association between that and Delta Smelt is weak at best and maybe non-existent.
That was very useful context. That would make what I said more clear. So I'll restate what I said in hopefully clear terms, which is using the current data and the current analyses, it's very difficult to tell if there's a relationship between X2 and smelt abundance. However, if we do measure these variables in a different way, perhaps measuring total production of algae, total production of smelt food, total amounts, total census size, that might lead us to gaining better estimates of whether there is a relationship between X2 and Delta Smelt. Hopefully that's clear.
Can I put one last point as well? Is that with X2, when people say there's no relationship, potentially no relationship with X2, it's not that X2 would never be important, because if you were to drive X2 out past the Golden Gate Bridge or to suck it right up the Delta, that could have effect within the kind of ranges people are discussing. There's not evidence for a strong direct relationship.
Thank you. One of the things that you may hear as we talk about going further in some of the other questions too, as a theme tonight, is about what we're calling a spatially explicit model. And what we're talking about in terms of X2 is that the influence of X2 on the environment and the organisms living there may be different in different as it moves through different subregions of the Delta system. And somehow that needs to be captured, and it hasn't been in the past studies. We'll probably get into a little more detail of this in some of the other questions.
We should keep in mind that all the studies of this system are observational studies, and they take environmental variables like X2, the salinity, or like ammonium concentration, or zooplankton concentration, and relate it to Delta Smelt abundance or the change in Delta Smelt abundance. These are correlations, and even if there are strong correlations, that does not necessarily imply a causal relationship. There can be indirect relationships, and lack of correlation does not necessarily mean that it's not important. For instance, one study did show a fairly strong relationship of X2 with longfin smelt, which is a potential competitor to Delta Smelt. So there are possibilities of indirect effects. We can't take X2 and manipulate it independently of all the other variables, do an experiment to find out exactly what the causal relationships are. So we have to infer them and take cautious steps.
So I just want to be clear with Dr. Lee's assessment. So you're saying that it's possible that if we measured food abundance, like copepods, in a different way, that we actually show there was a relationship with X2, which would then be at least an indirect effect on the Delta Smelt?
Yeah, potentially. Let me just put up a slide. I just want to put up a couple of slides to illustrate this point. Where's the adapter connection? was here a second ago. Disappeared.
What's right?
No.
Let's put it back here.
Do we have an AV person? Our AV person disappeared. Well, okay. So basically, if you look at Susan Bay, or a habitat like that, it's a large, shallow bay, and it has quite a large surface area. And if X2 moves upstream, then you're moving into a narrow channel. And I wanted to show a map that illustrates this shift in position, how it would affect the actual habitat. So there are several studies that have looked at the volume of the habitat of the low salinity zone, and the volume doesn't change that much. And those studies determined, or basically stated that with a change in X2, the habitat volume would not change that much. And they also, those studies also suggest that, no, it needs to be connected.
Oh, it needs to be.
Yeah, I don't know where the connection is.
It's right here.
Oh, I see. Okay, can you connect that? It's right here. Yeah. Sorry about that. Okay, so basically, as you know, low water flow leads to an increase in X2, such that the habitat of the low salinity zone will move upstream. And moving X2 might have profound effects that have been hypothesized on Delta Smelt abundance, but this is difficult to assess with existing data. So what I want to raise is a factor that the studies have generally not looked at. And so this is a low salinity zone in this area. It's the 2 to 6 parts per thousand zone. That's critically important for production of Delta Smelt. And this zone tends to be the region of highest productivity. It's also the estuarine turbidity max, where turbidity tends to be high. And this is where nutrients aggregate and where algal production, phytoplankton production, and zooplankton production, which is the prey that this Delta Smelt feed on, tend to be the highest. And this is where young Delta Smelt will congregate and feed, and then before they move upstream again. So basically, this is the copepod Erythrema affinis, one of the food items, and Pseudodactylum is forexi, which is another prey item. So with a change in X2, the habitat would then shift from this zone over there, that light blue area, which you can't see very well, to the light blue zone in the lower part of the Sacramento River, right in that region. Now, a lot of studies have focused on total habitat area. Actually, a few studies have. And they have basically stated that a change in habitat volume would not affect Delta Smelt abundance. However, one factor that might be much more important than habitat volume is habitat surface area, habitat area. And the reason for that is most of the primary production and secondary production tends to occur in the euphotic zone, which is the surface layer near the top. And the euphotic zone area tends to be correlated with habitat area rather than habitat volume. And so in assessing the viable habitat for zooplankton and the area available for primary production, that tends to be correlated with the area of the habitat rather than the total volume. So, sorry, I have a really bad cold. So if we look at some of the modeling studies that have assessed viable habitat, they have tended to use total volume in their modeling studies and in their assessments. However, I think that habitat area is something that should be looked at instead. And the other thing that they have tended to look at is the number of copepods, or the carbon of copepods per cubic meter or per liter. So they have tended to look at copepod density. However, copepod density tends to be very patchy. And the total census size, or the total amount of secondary production available for Delta Smelt production, would depend on the entire population of the zooplankton, the entire population of the copepod.
rather than particular density in localized habitats or localized sensing regions. So I think these are factors that are important and should be looked at. Sorry. Any questions about that?
Hold questions. Anybody on the panel want to comment on it? Okay, I think we'll go on to question 2. Since the 2009 seminar, many additional papers have appeared on the role of nutrient loads, nutrient stoichiometry, and contaminants on phytoplankton and zooplankton production. So the first question is this. Could concentrations of nutrients, such as ammonia, strongly affect food web structure, such as the production of phytoplankton and zooplankton, with a potential negative impact on Delta Smelt and or other species?
Does anyone want to comment on that first? Okay, so one of the key issues with nutrients is that different species of phytoplankton will preferentially take up different nutrients at different rates and have preferences for different types of nutrients. So a lot of the wastewater pollution, in terms of the nitrogen output, often is in the form of ammonium. And ammonium, for one thing, is taken up preferentially by smaller phytoplankton. So are most of you familiar with the spring bloom, the spring phytoplankton bloom? So those tend to occur through the large production of the larger phytoplankton, such as diatoms. And after the spring bloom, you then have a burst of secondary production by zooplankton that feed on that bloom. And then you get the production of smelt, young smelt, in places like in the low salinity zone. However, if ammonium concentrations tend to be elevated, they tend to inhibit the uptake of nitrate by phytoplankton, such as the larger diatoms. And in that case, even if the concentration of ammonium rises to about 4 micromolar, which is only about 1/10 of a milligram per liter, that can inhibit diatom production and prevent the spring bloom from occurring. And that would greatly depress the total biomass of phytoplankton in the system. So an increase in ammonium concentration could have quite a large impact on primary production, which in turn would affect secondary production and then Delta Smelt production. Is that all clear? Yeah.
Anybody else trying to comment on that?
I think that answer is pretty straightforward. There's also the issue, too, of the potentiation of harmful algal blooms by this.
Oh, yes, those are other issues. So one thing I'd like to mention, just to give you an idea of scale, the concentration of ammonium in wastewater that comes out into the bay could be as high as 11 milligrams per liter. And it only takes about 1/10 of a milligram in the system to inhibit nitrate uptake by diatoms that would inhibit the phytoplankton bloom. So 11 milligrams versus 1/10. That's a pretty large amount potentially in wastewater. So dilution of that through by having more water into the bay could greatly dilute that potential effect of ammonium inhibition. The other thing is ammonium is preferentially used by a few non-native species that are very harmful. For instance, the cyanobacteria microcystis is a species that tends to be toxic to zooplankton, especially the Copydrama affinis. And that toxic microcystis tends to preferentially use ammonia over nitrate. In addition to that, the invasive macrophytes, benthic macrophytes, water weeds, such as the Brazilian waterweed, also preferentially uses ammonia over nitrate from the water. And so a higher concentration of ammonium could not only promote toxic microcystis blooms, but also promote the growth of invasive water weeds. And these water weeds could affect things like water flow. It could affect the turbidity of the water, reduce sediments, reduce turbidity, and increase water clarity as well, which could be potential factors that affect Delta Smelt. others. So your ammonium is bad.
Given that, are the data sufficient to identify the likely ultimate sources of harmful nutrients such as ammonium?
It's clear from some of the literature that the sewage treatment plant or wastewater treatment plant has been identified as one source. The other sources seem to be non-point sources. And the data, what didn't make clear, at least to me, that the extent of that those sources contributed to the problem and exactly where they did. And one of my concerns for that, going back to this spatially explicit model again, is that we don't know much about the spawning event or the locations. We know kind of the general location. But it appears that those locations are up the river towards Sacramento rather than being down in the bays. And the presence of the ammonia, which can be toxic to eggs and to larvae, it raises the specter of an unaccounted for effect. So one wants to do the modeling so they take into account the different mechanisms that might be involved in this at different parts of the system, the river system versus bays and so on.
Okay, I think we've kind of covered question 3.
Yeah, that is my mistake.
No. Okay. So we'll move to 4. Since the 2009 seminar, a debate has emerged in the literature concerning juvenile entrainment of water export pumps as a mechanism which might result in periodic, at least, losses of Delta Smelt juveniles. Please comment on the strengths and weaknesses of the sampling methods and approaches reported by Kimmerer in his 2008 paper and the subsequent critique by Miller in 2011. So maybe somebody could review each briefly and compare and contrast.
The Kimmerer paper provided a first comprehensive analysis of proportional losses of Delta Smelt in the Sacramento River, as well as Chinook salmon. These losses were to water exports in the Delta. The Delta Smelt analysis used trawl survey data as the reference population. Miller, in 2000, I think it's now 2011, published a critique of the Kimmerer analysis. And I am in agreement with most of the points of critique of Miller. In order to estimate the proportional losses of Delta Smelt, one must estimate an absolute population as a reference. You have the fish that are salvaged, the fish that are counted. Salvages are the fish that are counted from the water that's being taken out of the system. There is an unknown number of fish that are lost in that process. So the idea was to estimate the volume of water corresponding to the fish that was salvaged and make that be the fraction of water that was taken out and correspond that to the fraction of Delta Smelt that were lost. The problem is you don't know where the Delta Smelt are in the water column entirely. They're very patchy as well. They're schooling, and whether you take this to be the volume of water from which you took this amount and counted the fish in the salvage, or whether there was only this volume of water from which you took this amount and counted in the salvage. That makes a dramatic difference in the estimate of mortality that results. The estimate of mortality
The actual amounts that were estimated by the Kimmerer paper ranged from 0 to 26% for juveniles and 1 to 22% of adults. So that's a lot of fish that are supposedly being trained at these places, and so that would be a very significant source of mortality if there's particularly on the higher end of those estimates. One interesting thing that the Miller critique did is he examined the assumptions that needed to be made to start the model that the Kimmerer paper used. So there were 8 of 10 assumptions that needed to be made that would lead to overestimation of the amount of information. So he went ahead and he did his own set of assumptions, and he came with estimates that ranged somewhere between 5 and 10%. So they didn't go up nearly as high. So, and like Brian, I too tend to favor the arguments that are made by the Miller critique.
The only point I'd like to make is that even if it is as low as Miller's just 10%, if the population is only experiencing weak density-dependent mortality, then it's still a 10% loss. So depending on the extent to which density dependence is operating in a population, if it's heavily density dependent, then losses due to entrainment might be compensated for by better survival later on, so they may not matter. So if they're not being compensated, then even 10% is a 10% loss.
I'd like to add that Miller had a response, Kimmerer had a response to Miller in 2011. And in his response to Miller, he conceded that his estimates might have been too high. There's quite a lot of uncertainty regarding information on distribution of the fish and efficacy of sampling and other factors. So he did later on concede that his numbers might have been a little bit too high.
For the modelers here, what do you need to be able to enter entrainment? What kind of data do you need to be able to enter entrainment into a model to assess its role in Delta Smelt numbers? Is there a way you could do it?
Yeah, there's a lot of different ways you can do it. The most obvious is to have estimates of the total population size and estimates of the number of fish in train, and just divide one by the other. Kimmerer took an interesting approach where he didn't have all the information he would like. So in terms of the abundance estimates, they don't estimate efficiency. So he just has a number of fish caught in a trawl, but he doesn't know what proportion of the fish are being caught, so he couldn't say what the total population size was, and he didn't know the efficiency of the entrainment. But he was able to factor those out in a way that I thought was a good model and a good approach, and it was very explicit with his assumptions, so he laid them out clearly, which allowed Miller to come along and examine them. And I feel like it's a good debate that's taking place there and kind of refining those estimates. So I don't feel like they're completely off track, and it would probably be good if a few other people entered that debate as well, so just having two viewpoints to kind of arrive at a consensus.
What do you think would be unambiguous evidence that entrainment caused the decline of Delta Smelt?
I don't think anyone's saying that entrainment has caused a decline of Delta Smelt.
Okay, what would be unambiguous evidence that entrainment is a major factor in the decline?
Again, I don't know if anyone can say it's a major factor in the decline. I think they're just saying that in some years it might be important.
This has been taken into account in some of the observational studies where they use the entrainment estimates as covariates to see how they stack up in relationship to other variables. The other kind of approach would be more of a life cycle type approach, where you model the various stages in the population as they go from larvae to juveniles to adults, and you explicitly would have a deduction in your population model for that mortality that's occurring. We've come up with estimates of other factors affecting the population at different stages, and then you could do a what-if analysis. If it was this much, how much does it affect the overall results of the population? And there's been some, particularly by monitoring the research, that is moving in that direction. The trouble is, there aren't direct estimates of the mortality components to be able to do that in terror, so I think that's still a result.
And the reason that Mondo Rizzo did find that the entrainment there was a weak factor as opposed to the other major factors that found that probably discussed under question 5. So the question becomes about this debate is if the entrainment data isn't as precise as we would like it, do we have to redo these kinds of models? Because if a new estimate comes in and it either flip-flops the position so that entrainment becomes more important or maybe even less important, that's important issues for us or anyone who's facing what to do about this problem in the coming years.
Although we don't have estimates of total population abundance of Delta Smelt, we do have an index of Delta Smelt, which fall surveys and others which rise when Delta Smelt rise and fall when Delta Smelt fall proportionally, reasonably. And at least 2 of 4 recent studies, extensive analyses of those indexes and the environmental covariance have shown detectable effects of entrainment on population level fluctuations, as well as a preliminary analysis that I did at the end of the 2009 seminar found an effect of entrainment as well. So there seems to be an effect, but beyond that, there is not a lot of knowledge.
Have you been able to extend that analysis from the 2009?
I did an analysis on the fly on the floor here in the building in the seminar, and I did a population style model. The panel had criticized previous analyses for analyzing Delta Smelt abundance rather than the changes in population abundance. The changes in population abundance are more related to births and deaths, the mechanisms, the biological mechanisms that are affecting Delta Smelt. And that's where the environmental factors affect Delta Smelt through effects on births and effects on mortality. So I had done an analysis on the fly looking at the changes in Delta Smelt and did an all variables model and found entrainment to be one of the variables that was most helpful in predicting whether Delta Smelt would go up or down next year. That data ended in 2006. A large spreadsheet of variables and abundances. The 4 big statistical studies that were published since then use basically the same data, and the data have not been updated. They're still in 2006, and it would be very interesting to take all the subsequent studies and get all the recent data which have not been collected together yet in one place and see which of those models actually predicts what happened from 2007 onward. That would be a fairly important test to put these models to.
Anybody else? Go ahead.
This brings up an important point about the modeling. The models, well, maybe this is a question for number 5, but let's just steal a little thunder since you talked about this. The models don't prove costs. They don't demonstrate costs. They help us understand costs. They help us understand what to look at in order to figure out costs, but they need some sort of field verification, like Brian has just talked about, in order for us to have confidence that they're predicting things in a way that we can have confidence that the mechanism and the model correspond to the causal relationships in the environment.
Anyone else want to comment on entrainment?
I'll just say very simply that the thing that bothers me the most is that the entrainments are not. They're measurements of other things that they eventually think would be a measure of entrainment, but they're not measuring entrainment as data. And that always makes me suspicious.
Okay, we'll move on to our next question. Since the 2009 seminar, several groups have applied more advanced methods and expanded data sets to explain Delta Smelt abundance. There are several new studies with the qualitative conceptual model by Baxter et al. There's a multivariate autoregressive model, a Bayesian change point analysis, a state-space multi-stage life cycle, and a multivariate hierarchical analysis. I'm hoping that we can have a discussion where you can comment on what these models, what perspectives to each of these models bring to the issues of water and Delta Smelt. And I'm not sure where to start. If somebody wants to take a stab at it, that'd be great.
Go ahead. I just want to say that these are significant efforts. All of these models are significant efforts, but they're not the same. They don't have the same approaches. They don't use the same sort of statistical techniques. One of them is a conceptual model. So they don't give you the same perspective on the problem. They each give you a little bit different perspective. And so one has to bear that in mind when you look at these. But as I said earlier, they don't necessarily predict or demonstrate costs, but they help us find out about what the cost is. Perhaps I'll leave it at that until we ask the next question.
There were 4, in particular, major studies published in the past couple of years that contributed new significant analyses to this problem. The Baxter et al. Paper is more of a synthetic review, bringing together all the possible information that's been published and summarizing it in a series of graphical models. It's a very nice place to really get started in the Delta Smelt and other story literature if we want to see all the things that are going on. But it does not contribute its own modeling or analysis of data in there. Instead, there are these 4 statistical studies, and they basically tackle the same data. A spreadsheet of environmental variables, all different environmental variables that are measured, and then these abundance measures. And they tackle the data in various different ways with various different advantages and disadvantages. The studies differ depending on whether they use a population dynamics model that looks at changes in population, or whether they do a simple statistical model which just looks at abundances and correlates the abundances with the environmental variables. They differ on what type of variability they assume for the populations. How much natural variability versus how much sampling variability. And they differ in the ways they attempt to estimate that variability and deal with it. That's very important because if you model the variability wrong, you'll get the conclusions wrong. There are different sources of variability in populations. The natural variability that would be there even if we're not looking. And then the variability that's reflected by the fact that if we go out and sample the population once and turn around and sample it again, we'll get two different numbers. The other factors by which they differ, these studies, they differ in the basic approach to statistics that they take. There are two very big different philosophies of statistics. One is called frequentist statistics and one is called Bayesian statistics. If there's lots of data, those two approaches converge on the same conclusions usually. But if there's moderate or sparse data, those two approaches will sometimes, maybe even often, come to vastly different conclusions. And what was my fourth? The fourth is, how many variables are they predicting in the model? The variables they're predicting are the response variables. One type of modeling approach just focuses on one response variable, say the change in Delta Smelt abundance through time. One variable measured through time. Another type of approach would take several variables at the same time. Changes in not only adult Delta Smelt, but the different other life stages as well, simultaneously, in a life cycle model. Or one could look at other possible interacting species, other pelagic fish that may compete or eat Delta Smelt simultaneously. Those data are available, and if one chooses to model those simultaneously with the Delta Smelt, you'll get kind of different answers. So these four studies all tackle different types of questions with different types of statistical analyses. And it's rather interesting that several of them come up with the same conclusions. There are three of the studies agreed that there's an effect of summer water temperature, water clarity, winter water exports, largemouth bass, which is a predator, July minimum Erythemora and pseudodiaphanous, that's the plankton food for smelt, and entrainments. Three of the studies agreed on water temperature. Two of the studies agreed on water clarity, winter water exports, largemouth bass, July minimum plankton for food, for smelt, and entrainments. And others were identified by single studies, included spring exports, calanoid copepods, inland silverside. Density dependence was very important. And other predators were important as well. The differences in approaches converged on several different conclusions about, or differences in approaches converged on some common conclusions about effects of certain variables. I think one should pay attention to those.
And yes, it's important to know that the kind of variables that they've converged on make sense. They have plausible mechanisms associated with them. And that's an important aspect when you're trying to judge the credibility of the credibility of the outcomes of it.
I mentioned earlier that the panel found in 2009 that a lot of the quantitative work was flawed because it didn't do the population dynamics correctly. I'm happy to say now that this has been alleviated, that this new work, for the most part, has been done correctly and taken into account the population dynamics correctly, and has therefore provided a much more solid basis for looking at the factors that affect Delta Smelt. As Brian mentioned, density dependence is one that seems to be among the most important factors. Now, what density dependence is, let's say between the number of spawners and the number of fish that return as larvae in the next year. What that means is that as the number of spawners increase, the number of larvae is also going to increase. But if there's density dependence, that increase is eventually going to level out or even reverse itself because of things such as competition, utilization of all of the habitat that's available, having large fish eat small fish, cannibalism, and other things like that. And so we now know that any studies that deal with these observational correlations need to also be taking a look at density dependence. I think that the other interesting thing is that in the life cycle work that Monder and Teresa did, they were able to look at density dependence not only between the adults and the larvae, but they also were able to see if it occurred between the larvae and the juveniles and between the juveniles and the adults. And so they ended up finding no density dependence between the larvae and the juveniles, that it was pretty much a straight line relationship, but that there was density dependence between the juveniles and the adults, suggesting that the biological effects on the population are more complicated than we have realized before. So I'm really excited about this new work, and I think there'll be more excellent work that comes out along the same lines.
I agree with Terry on the improvements on the modeling. There's 2 points I want to make there. One is a note of caution. All of the models are based on the trawl data. The trawl data is, in fact, everything we know about how the Delta Smelt has changed. The collapse has occurred is based on the trawls. And the way the trawl data is often, if not always, incorporated into analysis is quite naive. It just looks at certain areas, looks at either total numbers caught or numbers caught per volume, and it spans up. Even though we know spatially where the samples were taken, it doesn't take that into account. So you may have a clustering of points. You may have 4 points clustered together, and you might get a high count there. You might have 4 points spread out over here, and you get low counts on those 4 points. They're given the same weighting. So you would say, but in fact it could be all your fish are high density in one small area, and all this area here is low density. And so your numbers are lower when you take into account the spatial distribution of your points than when you just take them as independent points. And also the samples are done through time, and that's also not taken into account. So whenever it's justified, the point's always said, well, the numbers have gone down so much, it must have gone down a lot, and that's correct. There's no disputing the massive declines, but depending on how you analyze this data, if you were to take into account spatial and temporal variation, and also catchability, and I'll come back to that in a second, if you were to take all these factors into account, that might actually change the year-to-year variation, the small-scale variation, not the big trend, but just from one year to the next. And a lot of these models, a lot of the power of the models is looking at the tweaks in the year-to-year variation. So I feel like the analysis of the trawl data has lagged behind the analysis of life cycle modeling. And the fact that all the analyses are getting similar results, have to be borne in mind that they're non-independent, they're all based on that same data set. So I think it's important that data set goes back and is analyzed. And there's one more point I'd like to make. And you'll see this in the discussion of all the papers, this variable is important, that variable is important, this variable is important. Working as a consultant with clients, they don't want to hear it's important. They want to hear, if we change the flows, how will the number of fish change? That's it. People shouldn't have to delve into the science. They should just be presented with, if the flow was to change this amount, what would be the percent change in Delta Smelt? And that should be presented with a confidence interval. So that happens in one instance in one of those papers. And I'm not saying this paper is correct. It's in some ways we criticize it being one of the weaker analyses, but I just want to give you an example of the kind of thing I'm talking about. This is the Thompson et al. Paper, and they make a statement that their models suggest that an increase of 0.62 cubic kilometers in winter exports would be associated with a negative 22% decline in the abundance of Delta Smelt. So they've got specific output. If you would take this much more water in the winter, the model predicts Delta Smelt would go down by 22%. That's the best prediction, but there's always uncertainty in the model, and in fact they go on to say that it may be as much as a negative, it may increase by 45%. That's worst case scenario. But best case scenario, it may increase by 9%. So taking the flow in the winter could, according to their model, could increase the abundance of Delta Smelt by 9%. And this always gets glossed over in the discussion. It's always like, well, we should do this, we should do that. And to my mind, it's merging science, which is just saying, if you do this, how will the fish change? If you do that, how will the fish change? It's mixing the two up, and they need to be separated. So that, the best way to do that is via these models to ensure that the output is expressed in terms of which everyone can understand percent changes in Delta Smelt abundance with specific actions.
Yeah, I'd like to concur with Joe regarding the data, about the trial data, and I would also like to concur with Terry that the modeling efforts are quite nice. There's some really good models, really excellent efforts put forward. But what I see is basically a data problem rather than a model problem. And one fundamental problem that I see is a lack of linkage between the biotic data and the abiotic environment. There are a couple of studies, for example, that conclude that X2 is not a factor affecting Delta Smelt abundance. And as I mentioned to you earlier, it's inconclusive, in part because of the way that zooplankton and primary production is measured. I showed you a map earlier showing the area of the habitat, and as it shifts as X2 changes, the volume of the habitat does not really shift because that salinity zone volume remains the same, but the surface area changes, and the surface area does decline. And a hypothesis that I posed is as surface area declines, perhaps the total production, the total amount of zooplankton, the copepods that the smelt eat, might reduce in total census size. I mentioned to you earlier that what is input into the models that concluded that X2 is not a factor is not total amount of zooplankton, but the density, the number per liter, or the volume of the habitat, which also doesn't really shift. And these models, they do come up with useful relationships, such as prey density might be important for smelt abundance. But the ones that conclude that X2 is not important is using a number, as Joe mentioned, trawl data. They're basically going to a single site and measuring the numbers caught at that site. And they're not getting an idea of the total population integrated over the habitat space. And so, as I mentioned to you, that's a data problem, not a model problem. The models are useful, but the data going into them have to be relevant. And so I see a lack of relevance in the type of data that's being collected, specifically on the zooplankton.
To follow up on your comment about having an output that is useful for some or the Mondo de Rizzo model has some appeal because the model has processes embedded in it. It inherently tests for interactions. But because it also tells us where in the life cycle or the life history of the organism these particular effects are occurring, it helps us to be able to try to design some remedial actions. For example, the predation and the increased predation from the panfish, the bowgills, and crappie. That's something that could be doable in terms of a targeted harvesting program or something like that. You could say you want to decrease the level of those fish, which has gone up probably, 4 times in the last few years or something. I can't remember. But so you do that, and then you say, what does this mean now for the outcome for the Delta Smelt? Now, they're an annual species, so you won't have to wait tens of years, but you will have to wait some time. And so that's the kind of thing that allows you to use these models in a way that they call adaptive management, to make a change in your management practices, monitor the situation, and then figure out whether you did something that moves the system forward. And so that's why I'm attracted to Mondo and Rizzo's model, because it's this population-based model. It has the processes embedded in it, and it has the ability to inherently test the interactions. Some of the other statistical models only tested the main effects, didn't test for interactions. And so that begs the question of interactions in that case. To follow up on Dr. Lee's comment, when you look at the predation, food, water temperature, these are classic direct effects on populations, and not just Delta Smelt, other fish populations, indeed probably other organisms other than fish. So that's partly why they're very credible to us. But they don't necessarily tell us about the links between, well, they kind of say, here's the proximate cause, these changes. They don't tell you the ultimate cause. The ultimate cause is buried in what happened to change the prey field in the Delta system. And that's the second kind of model that's needed in order to understand what went on. And when you develop that kind of model, we need to know what went on where. What they call a spatially explicit model. For example, in the de Rizzo model, there were 9 factors that they identified, 5 of which have to do with larvae. And in people that study fisheries biology know that the period in a fish's life when they're coming off, they've been born, they've hatched, they've had the yolk sac, they've absorbed the yolk sac, they're now trying to feed for the first time. That's a very critical period in most fish's lives, and it's often the part that sets what they call the year class strength. How many fish are in the system that are going to be acted on later by these other things we're talking about. And that area in this situation between when the eggs are hatched, where the eggs are hatched specifically, and what happens during that yolk sac stage to the organisms, what happens during that critical first stage, what happens to them as they're migrating down the river to come to the base, is covered in de Rizzo's model, but in terms of the numbers, But what's not clear to us is the mechanisms that are involved in going from here to there. And getting at the mechanisms is going to be important to designing any kind of remedial action. I touched on the issue of the ammonia earlier. That's just one example. The whole thing about predation, as these fish are moving and coming to first feeding, is also important. So the water clarity part also means that two things to these fish that are feeding. They often need contrast. They have to see the prey sometimes against the background. So they often need particles in the water either to stimulate their feeding or to help with their feeding. What's going on? So what's the role of water clarity now in terms of the mechanisms that's affecting the Delta Smelt? Is it that? Or is it because under turbid conditions, their predators have a harder time finding them? If you know the difference between those two mechanisms, you can make some decisions about what you want to do to help alleviate the problem. And so I think you're going to have to now put as much effort into this kind of abiotic linkage, or this proximate ultimate in Dr. Lee's language, or this proximate ultimate causation in my language, in order to get at the nub of the problem. So, but frankly, I was surprised at a problem of this magnitude to look in the literature and just find out that they don't know where they spawn. They know about where they spawn, but they don't know exactly where they spawn. And so this is probably hard to do with Delta Smelt, that probably spawns at night and may not spawn where you're comfortable going in terms of a big boat or whatever. But you got to get at some of those thorny life history things that have been looked at off and on for years but never come to resolution.
Well, we've sort of been skirting this last issue, our refinements to the models needed. So what specifically? Dr. Lee suggested that X2 might be important, but in the summary that Brian Dennis provided, there are 11 things mentioned as direct factors in Delta Smelt, and none of them are X2. So what that suggests is that the models then need to be tweaked in some different way to perhaps test if that's the case.
Well, as I mentioned to you earlier, with the X2, it's a data problem rather than a model problem. The input into the models, they're using density data, numbers per cubic meter, or numbers per liter, or volume of the habitat. And that does not characterize the total census size.
Well, that would be a major difference to the models then, because none of them came up with X2. So that's an important thing.
But none of them use total numbers of cohort. They're all using the same.
So that would be an important refinement to future modeling.
Just to have data that actually characterizes what the prey abundance is, not the prey density. Because the density might be the same regardless of the habitat. But if the habitat is shrinking, the surface area is going down. If you look at the area of the habitat, it is shrinking quite a bit. And if you're only taking the euphotic zone, the top layer, its surface area of the habitat, then that's a several-fold reduction potentially. And if the total numbers of copepods, which is what they eat, is going down, potentially the change in X2 might be important, but none of them look at that.
Anybody have any other refinements to models that should be made?
The observations being used in these analyses only go back to the early 70s, only a few decades, and basically one each year is a different observation. So we have 10, 20, 30 some years. And that's not a lot of data points. In the data are something like 111 separate variables that are being looked at and tested for relationships with Delta Smelt. That's far more variables than there are data points. So there's not a lot of leeway to do anything really fancy with the data and look for more refined things, but I do have to remark that sometimes variables can show up not simply through main effects, but simply through their effects on other variables. The salinity X2 did not show up in all four models. All four of the big models agree that X2 did not have detectable direct effect on Delta Smelt. However, none of the studies looked at interactions between variables. An interaction might be the following, that the effect of ammonium, say, on Delta Smelt depends on the salinity. You wouldn't detect an effect of salinity X2 directly, but if you look at an interaction between it and another variable, you might see a strong effect. Such interactions are not present in any of these analyses. However, as you start adding on predictor variables, and each interaction is a new predictor variable, you start using up data very fast, and there's a limit as to how many of those one can look at carefully. But I'd like to see some exploration, at least, of interactions in future data modeling.
Right, thank you. Our last question has to do with the qualitative conceptual model of Baxter et al., in which they hypothesized that a regime shift had occurred in the Bay Delta estuary. And so what we'd like you to comment on is whether you believe the data and analyses support a regime shift, and if so, what consequences does it have for looking back and going forward?
Regime shift has got probably, well, I won't say as many meanings as there are scientists, but it has a lot of different meanings. And some people have very strict requirements for the phenomenon in order to solve a regime shift. Others seem to think that any kind of change might be that. But in essence, they have some sort of change in the state variables, the numbers of fish, for example, in our case, plus some notion that the factors that drive the system have changed in a way that they don't, there's a different set of factors now, or maybe the old ones that were there previously are working in a different way. And so that's the notion of a regime change. It's clear, to me anyway, that when you look at just the raw graphs of the abundances of the four fish that were looked at in Baxter's paper and then in the Thompson paper, that something happened in the early 2000s, and that it was more abrupt than the gradual decline that had been seen before. That was actually tested statistically by Dr. Thompson and his group. And the thing here is that it happened to the four fish with very different lifestyles at the same time. So that's a persuasive thing, that there were, whether you call it a regime shift or choose another name, that there was a change in the way the system was working. The other thing is that when you look at some of the graphs in the Baxter report, you can see that when he had colored the data points for those, I can't remember the exact figure now, when you color the data points for those years post-2002, you see that those figures, those dots were kind of flat, whereas the other dots were going up. I can't remember I apologize, I put too many graphs in this folder together. But that's another suggestion in the hard data that something was changing and that the drivers that were important in an earlier time in the Delta system are not acting in the same way now, or post-2002. So I strongly suspect that there was some sort of regime, or at least some sort of system shift. I'm also not going to dismiss the people that say, oh, well, you don't have enough data yet, because there hasn't been enough time since 2002. There hasn't been much time.
Anybody else want to weigh in on this? The environment is qualitatively different now than it was then, and what qualitatively different means?
I'd like to say that there has been a regime shift in the sense that ecologically the estuary is dramatically altered through all the invasive species, and we're not going back, not getting rid of those invasive species. So we're never going back to how the ecosystem was functioning historically. So in that sense, there has been a regime shift. There's also no doubt that there was a rapid decline early in the 2000s. And to the best of my knowledge, the cause of that decline has never been identified. And so, in all the discussions that take place about important factors and habitat being important, no one really often talks about what is the limiting factor. You can have all these things that would be good for Delta Smelt, would be good for a fish population. It's whichever one it is that's pushing the numbers down, that's the one that makes a difference. You could have not much habitat, but if something else is causing a problem, increasing the habitat isn't going to help. And so the general discussion debate really kind of talk in those terms. And I think that's important because saying, well, changing X2, is that actually going to change the fish numbers? And there's one last point I want to make which keeps slipping in and out of my mind. I have to wait to come back.
Then I guess the question is, how do we deal with the data?
Oh, it came back. And only on one point did I see it discussed that we have the 4 species have declined, and one of the species includes the striped bass, and yet we have large numbers of adult striped bass population. As far as I know, I don't know the numbers of striped bass larvae being trained, but whatever it is, it's seemingly it's affecting the Delta Smelt is also affecting the striped bass, and yet we still have lots of adults. And it seems to come on very suddenly. So looking at the data that way, it seems to be a specific unknown cause is causing these large declines. It's yet to be identified. So playing with X2 may cause small increases in numbers potentially or not, but I doubt any of those are going to cause an increase in the numbers back to historic levels until that cause is identified.
As far as what we should do, one thing I'd like to emphasize is, for example, in the St. Lawrence, where I worked with my collaborator, we actually measured total census size of the zooplankton prey for the rainbow smelt in the St. Lawrence. And the way you do that is not just by single point measurements, but you actually measure throughout the whole entire tidal cycle, multiple data points, multiple stations. So you can actually get estimates of how much zooplankton production, how much copepod production is going on, what is the total biomass. Because a lot of the numbers input into the model and a lot of the numbers that are being used to draw conclusions and regressions are basically single point data estimates that don't really capture what's going on. And a lot of the models suggest that prey abundance, the amount of zooplankton, is important for smelt abundance. Yet they don't measure. There are no measurements, as far as I could tell, no good measurements of zooplankton abundance. And that's a critical factor. And there's no linkage of that to where the habitat is, where X2 is. So I think so better data is something that we could really use.
So does this all mean that the model should consider pre- and post-regime shifts as independent estimates? Should they be analyzed separately?
Yeah, so this debate that's taking place here is if there's been a regime shift, what was previously controlling the abundance of Delta Smelt may no longer be the factor that's controlling the abundance. And that's the point I was making in the rapid decline of the unknown cause. Whatever that is that's acting may still be acting, and the other stuff, historically, most of the model is based on data pre the decline. What's identified as being an important factor there may no longer be an important factor. And another problem is, with the rapid decline, the numbers of Delta Smelt are so low, it's very hard to track the year-to-year variation in the numbers. You can say they're still very low, but the year-to-year variation, enough tools being done to actually quantify if those are real or just sampling variation. I don't know, that's not being looked at analytically. So in other words, we may have very little to no data on how Delta Smelt abundance has changed post the decline. And we don't know what caused the decline. And the factors that were important historically may no longer be important. So in short, we don't really know what's happening.
Having said all that, which I have to concur with, what is it they say, follow the money? I say follow the food. I think you need to do the kinds of things that Dr. Lee is talking about to tease out the details of the food situation. If there's no food, or the food quality has changed, which is one of the suspicions I have, then you can find that out and use that to make a better model. And the models you have, if the questions are couched right in terms of predictions that you want to make with the model and then go test it in the field, I think that still has some use. One of my biggest fears, though, when I saw that question on the document, was that in these days of budget cuts, they're going to say, oh, well, we don't need any of that trawl data anymore. Let's go off and do something else. And I would caution or urge some resistance to that attitude, because it's that abundance data that's been taken the same way, the same stations, consistently year after year after year, that allows us to understand that there was, in fact, some sort of change in 2002 and that sort of thing. Sorry.
I have two things to say about that. The first is just the technical issue of can we deal with that in terms of the models, or do we have to throw everything out and just start with the new data after 2002? Well, there's been quite a bit of work in the population dynamics literature that has looked at changes in productivity. A very good example are salmon populations. Salmon populations tend to go through these cycles, and they have definitely been affected by regime shifts. We know that very well in Alaska, because things have been great ever since the regime shift of 1977. But in places down here, they've been just terrible to see. At some point, that may reverse itself, or it may not. So anyway, there's ways of dealing with changes in regimes without having to throw out everything that's been done before, mainly by making the parameters of your density-dependent function time dependent. Second thing is, it's really been interesting to watch how regime shifts get translated into public policy. They get used on both sides of the field in different ways. Those people who want to get things listed under endangered species things say, oh my, there's been a regime shift. That means there's not going to be any habitat for polar bears anymore. They've got to be listed right away. So that's the argument that the regime shift should call for action under the UNESCO. Then there's those who would be on the other side who would say, oh, okay, well, there's been a regime shift, so there's nothing we humans can do about these things. It's the climate. We can't change the climate. therefore we don't have to take any action. Well, my point of view is the Delta Smelt population is in trouble, no matter what regime it's in. And responsible management says that you have to come up with some kind of mitigation measures to the best of our ability to change our human effects on the populations, regardless of what the non-human changes are. So I don't think whether there's a regime shift or not has that much to do with what we as humans do.
All right, well, for a bunch of 5 academics, there'll be only 10 minutes over time. That's pretty good. Our next schedule, calls for a 5-minute break, followed by a question period. So would you thank the panelists for me, and we'll open 5 minutes. Here's some pads of paper. If anybody would like to write a question, if you already have your own, that's fine.
Dr. Lee, is it possible that you could put our map back up when we do it?
Sure. They want the map back on?
What's the website that this is viewing on?
Website.org. In terms of where it's being located.
Where it could be watched?
We'll be posting it on website.org after.
Okay, but is it taking place now in real time?
It is. If you go to website.org, you can register and review it. There's a link to do it that way.
That's
Oh, you got to get through it.
If you'd like to ask your questions, this way.
This is a regular.
show here. I think that's part of the problem.
Is there a way to get
Yeah,
clarification?
it's because of the light. You see?
I see.
If they want to see it, then we have to read them.
Yeah, maybe we should dim the light, because there they can see the blue. Because I don't think this made any sense.
Okay. I meant that one there on the side. Can you just turn that so that she can see that one? Yeah, turn it so it's there.
Yeah, because the microphone is here, but not as we went further, so we're trying to add some.
Oh, so you were able to hear me?
Yeah.
questions. This is good. This is good.
Bob, great job moderating.
Pardon?
Great job moderating.
Well, at first I thought we were going to be done in 5 minutes. And so I kind of let it drag out, and then I realized, whoa, we got to reach.
Okay.
Thanks, you guys do a good job. Let's keep it rolling.
Amplify my thoughts. You rolling? Okay, well, I can answer the first question. There is a 99% probability that the Minnesota Vikings will lose every game this year. All right, the first question is, could you please provide some information on the possible role of the introduction and explosive growth of the Asian clam on the Delta Smelt population? Anybody want to tackle Asian clams and Delta Smelt?
When we go back to this water clarity or turbidity factor that was used in the models, the Asian clam could be, I don't know for sure, but it's a filter. It does in fact clean up take the particulates out of the water. In other places where invasive clams have come in, they've cleaned, like Lake Erie, they've cleaned up the east or western part of the lake to such an extent that the fish populations have indeed changed. And visual predators, which were handicapped under the high turbidity, where there was highly turbid water, can now see their prey much better, and their populations have increased, while other fish populations have decreased. That a similar mechanism could be going on with the clams that have invaded the Delta system in terms of changing food clarity.
I'd like to add that the invasive clam corbula also eats copepod larvae. Copepod are the zooplankton food source prey item for Delta Smelt. However, for the clam corbula in particular, its salinity ranges from 1 parts per thousand to about 30 parts per thousand or so, but it is more abundant above 5 parts per thousand. And the range of the copepods and Delta Smelt is the blue range up there, the low salinity zone. So the clams are more abundant in the slightly higher salinity zone, but they could consume a lot of copepod larvae where they do occur.
This goes in a very different direction. Should X2 be totally based on water from only the Sacramento River?
So what does that exactly mean?
It can't. The way X2 is defined is the distance from the Golden Gate Bridge, or the Bay Bridge? Golden Gate Bridge to
parts per thousand.
the 2 parts per thousand isohanging. So you can't, the Sacramento doesn't come into that definition. The flow determines the position of X2 or the value of X2. And that's a different proposition.
Okay, this one's more specific and maybe hard to answer. Does tidal influence during monthly high tides change the smelt entrainment to the pumps in Clifton Court 4 Bay?
Can you state the question again?
Does tidal influence during monthly high tides change the smelt entrainment to the pumps?
Yes. The pumps are way upstream in the freshwater area, and I don't know what the tidal influence is at that point in the river estuary system. It's certainly influential down in the estuary portion, but way upstream, I don't know that you have a lot of tidal influence at all.
There still could be some because you can get tidal influence far up a river even though the salt water is down at the mouth of the river. But you're right, the further up the river you go, the less that will be. What were they asking for, spring tides?
Monthly tide?
Monthly high tides.
There's the spring tides are the highest tides of the month. So conceivably, those are the ones that are going to have the most reach up the river. There's data you can check to answer that question, but I don't know what we have here right now.
So is the potential consequence that you would reduce exports during those particular high tides if Fresno was an issue?
I don't know whether they're saying that the high tide will bring more fish to the pumps or lessen the problem.
I mean, often these types of questions we're not able to answer because we're not very familiar with the systems like the people who have been working on them for 20, 30 years. Kind of where we come in is we're able to assess the science and spot inconsistencies and general problems and point out general directions, but specific, intimate knowledge of the systems. We have 3 days to prepare for the meeting, so we don't really know.
Let's try one more of these. Would an increase of water from the Sacramento River to the San Joaquin River upstream from the pumps benefit the smelt? So would an increase of water from the Sacramento River to the San Joaquin River upstream from the pumps benefit the smelt? Something like that. Get it over.
Does somebody have a question about this? Somebody requested me to put up this map. Yes, did you want me to?
No, I just think as we're having this discussion, I mean, I think some of these questions
Okay.
perhaps are not fair to be asked if you're not familiar with
Oh, I see, yes,
how the
that's a good
state
point.
and federal pumps operate
Yes.
in concurrence with one another. For example, that last question, I mean, the San Joaquin River flow is much more de minimis than the Sacramento flow for a number of different reasons based upon how the water is appropriated. And the Clifton Court 4 Bay is down, keep going down, up, over this way here, down further, we're getting into the Delta, further
Yeah,
north.
this is the Sacramento. This is the
Now, right about there, where it doesn't show it on the map, is where the state and then the federal pumps are.
About in here?
Right, yeah, about there. And so, I mean, if the state and federal pumps at the summer or toward the fall on low water years are all operating ridiculous. It's 12,000 CFS plus, I think 13,000. The state pumps are 8,000 CFS, and the federal pumps are 4,500 CFS. You can actually turn in the direction of the water flow. The pumps are powerful enough to do that if you have low flows, very low flows in the late summer and in the fall. And that's one of the reasons that they don't at different times. But I think it's hard to take that into account. Some of the other people here probably have more familiarity than I do in terms of how the changes in pumps operate over the course of the year. So I'm not so sure it's a fair question to ask you in terms of that. That is, the X2 factor that we're talking about, we were talking about, where Judge Manson was principally go in effect 2 weeks ago of September and October, which was when the state contractors get a large percentage of their water. And that's why it was a big issue because of how much water, the additional water that they would have to take out of the state project to reduce those salinity levels down to the points that you're talking about. And as a result of the decision that took place here 10 days ago, and it was upheld, means savings of anywhere from 250 to 400,000 acre-feet of water during this 2-month period of time, based on the X2 factor and how the court ruled. And I don't know that any of that is applicable to what we're discussing.
Well, to my mind, what goes through my mind in response is that, boy, I wish we had a spatially explicit model right now.
I guess my question, I don't know if it's appropriate, but at some time when it is appropriate, I would like to ask the question.
So I guess we're going to punt on that one too. There are two questions that are similar, and they have to do with identification of a number of factors or stressors in the system, and they include food for the fish, flow, export pumping, predators, upstream diversions, wastewater discharges, and so forth. And the questions both kind of ask whether you would care to attempt to prioritize them or identify what you think might be the biggest threat. How would you respond to that?
One of the advantages of two of the approaches that were used was they tried to get at just how much each factor plays a role in the population dynamics. That's in the one of the original model and in the locality.
Which one?
Which one? The McNally model? Right, but I think there's one of the models had how much percentage of variation is explained by the different factors. So we don't have the answer for you right now, but it is available through some of the analyses that two of the models did, I believe.
Can I make a point from that? So often when the models talk about importance, and this is important, or this explains so much of the variation. And you can explain a lot of variation, like if you vary a lot through time. You may not vary very much through time, so you wouldn't explain much of the variation through time, but you could still be quite an important variable. So that's where quantifying the output, as the point I made earlier, as if you were to change, if you were to reduce striped bass numbers by 50%, what would be the percent change in Delta Smelt? If you were to remove water weed, what would be the percent change in Delta Smelt? If you were to decrease flow extraction, what would be the percent change in Delta Smelt? Every one of these variables needs to be quantified in terms of a change in that and how that would result in a change in the Delta Smelt. So simply saying, like doing an experiment with a drug and saying this drug was not an important variable, when everyone got the drug, there was no variation in the drug levels to actually see a difference in the response. Does that make sense?
Yeah.
So So instead of saying it explains this percent of the variation in the data, actually saying if you change it, what will be the percent change if it does not actually get what's of interest?
So the one about the McNally, I stand corrected on the McNally study, but I think that Monte Rizzo model still has the potential that you're speaking of.
My understanding, they look at impact, and that's saying if it wasn't varying, what would be the importance? What is the impact if it wasn't varying? But it still is confounded with the fact that a variable that varies a lot through time is said to have more impact than a variable that's changed very little through time. But that variable that's changed very little through time could actually result in a bigger percent change in the Delta Smelt. So I don't think it completely gets around the problem.
Doctor, can I ask you a question?
Yeah.
If I followed you correctly, because we don't attempt to quantify those variables on the other stress factors, that would lead me to believe that is a underlying flaw in the science that's being used to try to determine as to what impact, whether we're talking about X2 factors or flows at other times of the year. The X2 was just for the months of September and October, but other times of the year, flows are being used to make a determination as to the health of the Delta Smelt, and then how it impacts the rest of the food chain. If we can't quantify the other stress factors, it seems to me to be a real significant issue as to whether or not you can make any facts or any determination on flows to either increase or decrease the Delta Smelt.
To respond to your question, I think there are maybe two parts of my response. One is that I don't see that science is necessarily flawed. It's just the outputs are not in the format that is useful for decision making. And this is because a lot of science are academics and they're interested in academic questions and generalizations and methodologies. But having been an academic and now being a consultant, I know the clients want it in a certain form. They don't care about the science. They simply want to know if they do this, what will happen. And these are papers published in peer-reviewed journals, and the criteria for acceptance and the formats are not the general format that's useful for management purposes. But the science underlying them is generally very good. So it's just outputs. And
well, maybe another way to phrase it is you're not taking into account other elements that will have an impact on the smell.
Yeah, I mean, you can take any model and you say, well, let's hold all these variables, let's assume these variables at their current levels, and let's change this one variable. What does the model predict is going to happen to Delta Smelt? Okay, great, so we change predator numbers, but we hold everything at the current levels and we see what the model predicts would happen. And then we say, well, we're not going to change predators, let's change this variable, let's change flows. Hold everything at current levels, how does it respond? Okay, let's try and change two of them at once, let's see how it responds. So those model output, I do that type of modeling all the time. Those models just need to be, the outputs need to be changed into a different format that's directly applicable to these types of questions. So you could quantify what the model predicts reducing fish predation would be, what the model predicts having more coconut pods would be, etc., etc., etc. But it's just not in a format that's useful for making decisions.
I think you're asking about the next step that the modeling should take, which is to make the predictions that he's talking about and then verify them in the field.
Yeah, may I ask a question?
I have a response. All of these factors that we've been talking about generally have been quantified to some extent, and all of those variables are brought into these analyses. But what is done is you can't put every single variable into the models because all these variables are interrelated with each other, and so you just end up with a whole bunch of garbage if you stuck them all in there. So what scientists do is, for example, in the Monger-DeRiso case, they put in the density-dependent parameters because those have already shown to be very important. Then they take a look at the two most important factors that seem correlated with the change in abundance. And then after that, they'll take another two, add those in, see how that affects things. And then essentially using that in conjunction with fitting criteria, they'll come up with the model that best explains the data, but still has the fewest number of parameters necessary to get the best explanation of the data. So it's not that they're not being considered, it's that they don't show a relationship with the change in abundance.
The members of the panel have considered the various models and discussed them tonight, but the question I'd like to ask, and do those models contain the stressors that are thought to have the highest probability of impacting the Delta Smelt?
Well, the answer would be no, in my opinion.
Right. So,
especially for prey. I mean, prey density, numbers per liter, is not a good indication of the total amount of prey available. It's a very bad indication. So I'd say absolutely not.
in my experience with modeling is that you can do multivariate modeling with your independent variables being, in this case, would be the various stressors. And looking at the variances associated with each one of those, you can come up with a better summation. If, of course, you've chosen your dependent variables properly and your data sets. Those are all kinds of qualifiers. But if you can come up with some idea of what is the more important independent variables insofar as the impacts that you're looking for, am I correct or wrong?
You have to, and this is, you have to quantify when you say most important. Most important with respect to what?
Well, in this case, we're looking to the health of the Delta Smelt, and so you'd be looking at things such as ammonia, possibly the clam, flows, and I'm sure with a little thought we could throw a few more in.
And the current batch of models is pretty sophisticated and looks at a lot of different variables. I generally don't have much of a problem. I think the modeling is pretty best available science. I just have some problems with the inputs in terms of the fish numbers not taking into account the spatial and temporal non-independence in the variation. And I have a problem with the way the results are presented. There should be the percent change in the Delta Smelt and the change in each of the variables. But generally the models are pretty good and they include a lot of different variables. They're limited, but they're pretty state-of-the-art.
Yeah, I agree. It's not, modeling itself seems to be quite sophisticated. The problem is the way in which the data are collected and the type of data that are put in is problematic.
I'll add, there are these two separate questions. One is the data quality, and the other is if you rely on the data to make predictions about the Delta Smelt index, what predictions will it make and are they any good? On the prediction side, these modeling efforts have typically taken enormous computational approaches, fitting all possible subsets of predictors to see which of all possible subsets give the best predictions of Delta Smelt. And those are the variables that I mentioned and we've discussed that emerged as important to Delta Smelt abundance from these statistical studies. A small handful seem to do the best job of predicting Delta Smelt. And you can take for each one of those variables, you can go into the model and look at a coefficient, and that gives the predicted increase or decrease of Delta Smelt per unit increase in this variable, such as entrainment. A unit increase in entrainment will be predicted to make this amount of difference in the growth of Delta Smelt according to these models. The predictions of these models should be tested now with the new data when that is assembled since 2006, as well as some possible experimentation. I would encourage the managers and the stakeholders and the biologists in this system to take a broad view of experimenting in the system and do some system-wide manipulations. This is a process that's called adaptive management, where you take the results of your management and make new hypotheses and use management to learn about the system. And I've encouraged the managers to be very profligate in giving the system some probes, some water stresses, and some other stresses to see what happens. We're not going to eliminate Delta Smelt immediately. We can endanger them with long-run stresses, but in the meantime, by doing some well-designed manipulations over a period of 5 years or so on, you can learn a lot about this system.
I'll just give an example with the Monte-Teresa paper. When they collected the number of variables they had 24 different variables that they considered across the 3 life stages in their model. And these variables represented various aspects of factors related to things like temperature, water, clarity, and things like that. When they finished their analysis and eliminated those factors that didn't seem to have any explanatory capability, they still ended up coming up with a model in which different covariates affected different stages. But the covariates that were in the models included temperature, prey, predator, adult entrainment, and water clarity. So these are the main stressors that we've been talked about. I think what Carol's point is that there might be better variables that can be constructed out there that have a better biological and ecological basis. And we've seen that that's true not only for the independent variables, but also the way the data on the Delta Smelt themselves are processed. There's probably improvements that can be made into how to standardize that to be a better measure of abundance.
Yeah, I'll be more specific. Maybe I'll clarify what I said. If you look at that map right here, the one above is a smaller X2 of 74, I believe. The lower one is a larger X2 of 95. And in the Monte-Teresa paper, they use habitat volume or copepod density times volume to estimate prey. And in their population model, they do find that prey abundance is very critically important for Delta Smelt abundance. In fact, several of the models concur and come to the same conclusion that prey number is important. What the studies all find, though, is that X2 is not important. But one of the problems with their assessment of that is the linkage between, when I say linkage between abiotic and biotic, the linkage between the prey amount, the zooplankton amount, and the habitat is poorly done, because what they're doing is they're measuring the number of copepods per volume, or they're using total volume of the habitat. So as you go from X2 is 72 to X2 is 95, the habitat volume actually doesn't change. What is changing is the area. And if you see from the map above to the map below, the area is changing. And that's what's critically important for photosynthesis. The area of the habitat is what dictates the amount of photosynthesis, which dictates the amount of algae, which dictates the amount of zooplankton. And in none of the papers did I find an accurate estimate of the total amount of zooplankton. There's estimates of volume, there's estimates of number per volume. And so that is a basic piece of information that's available for other estuaries like the St. Lawrence. We have a census size of zooplankton for the St. Lawrence. It's 10 billion, approximately, copepods in a particular season. We have those estimates for other estuaries. We don't have this very, very, very basic number of total zooplankton synthesized for the San Francisco Bay Estuary, as far as I can tell. And that's a critical number. It's the total amount of zooplankton that's going to give rise to the total amount of smelt, not the density. Is that clear? Did I make that clear?
I guess then these questions all kind of wanted some sort of conclusion, and we certainly identified the number of factors that are present in three of the four studies, two of the four, one of the four. So we've ruled out maybe a class of things, but this question leaps out: what remedial actions, if any, do you recommend? What scientifically has to be accomplished before the science will be able to actually make these remedial recommendations, and we don't have to refine the models any further? When will we be there to the point where the science can make, clear guidelines to managers on what to do? What do we need? What's the cheapest way to do it?
In my concern is if you take the original model right today and look at what it might predict, what you would do it. If you reduce predators in the system by half, what kind of effect on Delta Smelt would you make? You can call up Dr. Rizzo, ask him to do that. You'll get an answer in a day or two. And then it's up to the fishing regulators to design a targeted harvest program that will remove the panfish. And then you'll see the result in a year or two, probably 2 years, just to be safe. You can do that today.
Could you do the same with ammonia?
Yeah, if it's been included in the model, you could run the model and say we're going to hold everything at constant levels, and if you were to change the ammonia, what does the model predict would happen to Delta Smelt? And you could see what it tells you. You could and you should do that for all four of those models. And you should look at each of the variables and you should ask which of the ones can we change and how much will it cost, and say what's the cheapest way of changing Delta Smelt based on these models, based on the best available science to us.
You don't even need to hold things constant.
Well, when I say hold them constant in the sense of current levels, whatever they happen to be currently. If you have interactions between them, the effective value depends on the level of something else. And because you don't just want a relative number, you want an absolute number so you can get a percent change in current Delta Smelt levels.
Bob, if I could take this in another direction, slightly different direction. The Fish and Wildlife Service is going to have to sit down with the Bureau of Reclamation and the stakeholders, and they're going to have to develop an opinion as to whether or not the way the Delta is currently operating is likely to cause jeopardy to the Delta Smelt. And the question, I suppose, is, given that their prior opinions fell short of the necessary standards, and there's additional information now, if you were the person in charge of writing that biological opinion from the Fish and Wildlife Service, where would you start?
I'm not sure if this answers your question, but if I was in that person's shoes, I'd be on the horn to whoever did the entrainment studies to find out more about the estimates, and I'd replug those estimates back into the Rizzo model and see what that told me about the influence of entrainment versus all these other factors. If it was still a significant factor after I got the new improved entrainment models, I'd start to play what-if games. If I increase entrainment by X, what happens? If I decrease entrainment by Y, what happens in terms of the model output? And that You can do that entrainment what-if game right now with the model that's present. The issue is whether the data that was put into the model is the best data that we could have on the question. Similarly, the question on the food is you can play with that information right now, and you can get an output. But the question is whether the information that gauged food was complete or not, or was done in the most appropriate way. So you can get new data and do that. But the problem with the food is that how are you going to change the food system? And that's the problem with the ammonia. You can probably make the case that we should be doing something with ammonia anyway, and maybe this thing needs tertiary treatment or whatever. I don't know what the status of the system is, but trying to do the what if games there, it's not clear to me what the management action will be to increase the food supply, either in level or quality.
Well, actually, one back of the envelope thing we could do, I mean, the best thing would be to collect census data on how many zooplankton copepods are out there. But you can do a back of the envelope calculation with the X2, for example, because we still don't have conclusive answers about the potential impact of X2. You can take the habitat area, you can calculate the volume of the euphotic zone where primary production photosynthesis occurs, and look at the area of where the low salinity zone is, and do a back-of-the-envelope calculation of how much of that area is reduced when X2 changes, and get a very, very, very rough estimate of what change there would be in copepod abundance, zooplankton abundance, just through back-of-the-envelope calculation. So under different X2 scenarios, how would the habitat area shift? Because habitat area is likely to be correlated with zooplankton abundance. Very rough, though. So that's one estimate you could get. The better thing would be to go out and measure copepod census size. But that takes a lot more work because you need to do it over multiple tidal cycles.
So
and census better.
you seem to be saying that right now you don't want to have to make a decision. But what if you do have to make a decision?
You work with the tools you have.
We can do a better job of working with the tools we have.
Yeah, but on that point, on the tools that you have, and I don't know if there's a consensus among the scientists here or not this evening, that the models seem to have a level of integrity. At least that's what I think I'm hearing from you.
Yes.
However, there also seems to be a consensus that the data that has been put into the models, either by the way it was collected or sampled or processed, has not necessarily allowed us to make determinations because of the poor quality of the data. Am I making an assumption here that there's a consensus there or not?
It's fair, except that you can still run the model as it stands.
Yeah, but isn't it?
But you don't have, we wouldn't have the faith in it.
Well, to my point, garbage in, garbage out. I think that's another analogy. But that's still, if the data that you're putting in is not good data, then you can have the best model in the world, and it's not going to tell you that.
Yeah, and the results may not change, but I think, in my personal opinion, it needs to be rerun with those inputs before you can say it's based on the best available science and data. And then the what-if game has to be played with the outputs based on that data, current models, the outputs to look at percent changes in Delta Smelt, the changes in different variables, and then you need to make your decisions about what you're going to do.
If I were the science manager of the system, probably ask for three things. One is to collect the data that exists and check all these models for their predictive ability since 2006. Second, I've launched a data audit of the practices of data collection and data processing to see if those could be improved to be more biological relevance. And I would especially be happy if some of these back-of-the-envelope calculations, so-called, could be retrofitted onto the existing data so that we could have these trends since the early 1970s and be able to continue to follow those in some way that would be using the data that we have but processing them so that they give more meaningful information, such as combining copepods and X2 together to get some kind of estimate of overall copepod abundance. That's sort of like looking at an interaction between X2 and copepods in the models, which we have not yet done.
Are you scientists familiar with the term dilution as a solution?
Dilution is the solution.
Yeah, the more water you flush out there, you can cure all the ills.
Yeah, the solution to pollution is dilution.
Right.
Okay.
That would be true for ammonia, probably.
That's approximate, or I'm going to say that is for the ammonia issue, if that's what you're speaking to. That'll work to a certain extent, but then you still haven't done anything with the source term, the source of the ammonia or ammonia, and I submit that if you have to manipulate the water rather than just fixing the thing at the source, that brings in all these other social and economic issues that face the community. But I'm losing my train of thought here, whatever it is.
I'd like to add that just a point of information, in regards to the ammonia discharge from the Sacramento Regional Sanitation District plant, with their permit, which was imposed upon them in December of last year, they're required within, I think, a 10-year period, if I'm not wrong, to go to tertiary treatment. In other words, they're having to remove the ammonia. And so that one stressor, at least from that plant, and I might point out that they, I forget the exact numbers, but they were the source of roughly 40% of the potential ammonia that could be going to the Delta.
Really?
Most other plants have gone to tertiary treatment and are not contributing to the Delta. They're the last major source of ammonia to the Delta, and that will be in time removed.
So the last major point source.
That's correct.
If there's the non-point source, do we have an estimate of how much ammonia comes from the non-point sources?
There is an estimate, and I don't have it. It's basically upstream in the agricultural areas.
How about in the urbanization around the Delta?
There's always contributions from septic tanks, from
Lawns?
well, there's pesticides coming from the lawns, interestingly enough.
My question is fairly simple, I think. Given so many variables, and given that this is a commercial area that now has got environmental impositions, with fish and everything else, have any of you ever seen this successfully studied and corrected in other real-life situations?
You mean related between fish and flow?
Well, all the variables that, you've taken a commercial area, and now you apply environmental concerns, and now you study it. Have any of you had experiences where you've been able to study and correct it and actually come out on the other end successful?
I was going to talk about Wisconsin. I'm from the state of Wisconsin, and flow is a huge problem with dams. And there have been several studies that found that the removal of dams and increase in flow has improved the integrity of ecosystems. So there have been a lot of studies. that are analogous to this in Wisconsin.
But that's just flow. In other words, you've got
Well, flow was a big factor in that case. So in that particular example, flow was a huge problem. That was corrected, and it led to improvements in the ecosystems in those particular cases. And there, flow was the big stressor.
I was going to say, the systems I've worked on, dam systems, definitely we've improved our knowledge of being able to adjust the flows to improve fish numbers. So it can be done. This is undeniably an extremely complicated system. To the previous question, to something about almost implied that we ever have complete understanding. I think we absolutely will never have complete understanding of this. But in the end, as far as agriculture is concerned, the question is what's the relationship between the fish and flow? That's it. How you change the flow numbers, water extraction, how do the fish respond? So we have the existing models that need to be improved with making sure the data going in is of the highest possible quality. Then they need the outputs to say, what if we did this with the flow? What would happen? And then you need to be monitoring those Delta Smelt so well, when you do change it, you see what happens. So you need to ensure that the current levels of surveying is sufficient to give you a handle on Delta Smelt variation at very low levels. It may be that a lot of research money is being channeled into research and other causes which you may never fully understand should be done into extremely good trawling. So you can say, we know exactly what's happening with this fish. We're going to change X2 to this level. We're going to see what happens. Okay, we're going to take a hit and we're going to change it to this level for 3 years, and we're going to see if the Delta Smelt responds. And if it doesn't, we're going to push it over here and we're going to see what happens. And in that way, it's got this incredibly complex system. You're just taking your two variables of interest and seeing if you can relate the two of them based on the models of the what if and then doing the true what if. And the systems I work on, and something that gets so complicated, I just fall back on making sure I know exactly what the fish are doing, and then we tweak the flows and we see what happens. And that way, in the end, it's just a win-win situation for everyone, because you know what's happening with the fish, you know what's happening with the flows. And that may be your best attack on the problem.
Yeah, but it is a very complex system, as we all know, and agriculture, from our perspective, from my perspective, is essential. But this plumbing system, this is the linchpin of a water supply for 25 million of the 38 million people that live in the state. So, which just further underlines the complexity and therefore the importance of making this plumbing system work effectively for a stable water supply for all that depend upon it, and still deal with these issues of fisheries and other environmental related issues. So I mean, I just think that, and we're not even taking into account the potential impact of the change in climate, increasing of the water levels a foot maybe in the next 50 years.
I just wanted to mention that there's several other examples that point to the hope for success. The Lake Washington system in Seattle is probably one of the best examples. In the early 20th century, it was just a quagmire of pollution, and they spent a lot of time cleaning that up, and now it's pretty much a pristine system. Another one is the Duwamish Waterway, which is the most industrial area in Seattle. They're creating a whole natural park system along the river there and restoring it to pretty much pristine condition. And then, of course, the Ohio River that went caught on fire. They've done the cleanup on that too. So yeah, this system has experienced a lot of insults, but I think that with wise choices, that there's no reason that you can't make things better.
To respond to your question there, Jim, I think science just really deals with, if you did this, what would happen? What you're kind of discussing is the importance of the water and kind of policy decisions. But as a scientist, I just restrict myself to, well, if you did this, what will happen to the Delta Smelt? and then just trying to be honest and saying the extent to which the uncertainties are there, which is really expressed in the percent change in Delta Smelt, which could be positive or could be negative. So the science is really should just be, if you do this, what will happen? If you do that, what will happen? And then the policy is trying to find solutions to within that framework.
So could we write a biological opinion that would not be considered arbitrary and capricious?
Is that what the question?
Yeah.
Not with some more work, not with some more number crunching on the data to make sure the data is absolutely right, redoing the models, making sure the outputs are looking at all different variables, percent changes in Delta Smelt. At that point, we could probably sit down and summarize it the best we could, and the conclusion may be that we don't, but we'd see what the models gave us. At this point, it's impossible to say.
With the fact that the X2s up there that you have, all the water that pretty much maintains X2 comes from the Sacramento River, because the San Joaquin River has been significantly dewatered, and it continues to store water all times of the year, including the driest time. So you have basically the same amount of wastewater that's being discharged into these waterways, but you have a diminished flow that you're putting that water into. With those concentrations, when you look at the X2 and moving it further upstream from Golden Gate, you're going to significantly decrease the amount of water available. It will increase the water quality. But at what price? And we're probably 5 to 10 years away from the tertiary treatment plants being finished. So we're going to see a continued diminishment, I think, of the water quality of the Delta in particular in the September-October period of time, because there's just not enough water flow to dilute the wastewater, which is basically the same amount all the time, regardless of how much fresh water flows. So I don't know if that correlates one against the other, gives you the concentrations, it gives you the toxins in the Delta, but we're moving in the right direction, I think, but we haven't got there.
Do you know how much ammonia is coming out of that one plant?
About 17 tons a day, I think, is what I've heard. And I have some papers that I'm pulling up now that I'd be happy to share with you. There is really mixed evidence on the impact of the ammonia on the Delta Smelt, but it is pretty evident that it is impacting a number of the invertebrates. So its impact may not be direct, but certainly would appear to be indirect.
Well, it definitely would impact the phytoplankton.
Exactly.
Yeah.
And there are some investigators who feel that is the more probable
Which would then
outcome of that
right. Which would
discharging.
affect the food supply.
Well, to give us more information, last year it was estimated that tertiary fix would be somewhere in the neighborhood of $2 billion. So how that money is arrived at between the users of the treatment facility and whether or not they get any state or federal money, certainly sewage rates would have to increase dramatically to pay for that fix, but they're now under the water quality control board. They're given a timeline.
That's correct. Actually, they estimate $2 billion. The water board staff estimated $1.2 billion. A lot of money, but still a significant difference. They have other opportunities that over time may to some degree mitigate that. Certainly that water has value. Certainly they have declining groundwater basins, overdraft upstream. Some of that water can certainly be treated and used for recharge. So there's savings in other arenas that they might realize. It's going to take a large study to figure out how they're going to address this, but certainly they do have options that can, to some degree, mitigate the financial impact that they will be facing.
Well, I think the task of sorting through variables, identifying relative contributions is a Herculean task, to say the least. Do any of you have any last comments you'd like to make?
I think the task has largely been done. It's just checking the data inputs as good as they could be and making sure the outputs are presented the right way. But a lot of the work on the modeling and looking at the variables, a lot of it has been done.
Well,
It's good to hear some good news.
the Fish and Wildlife Service was due to publish a draft biological opinion tomorrow, but this afternoon the court entered a new order. The parties have agreed that they will agree to a new date by October 30th. And if they don't agree to a new date by October 30th, then the new draft biological opinion will be due on November 11th. So people are scrambling to make these decisions and come to some conclusion. The Fish and Wildlife Service and the National Marine Fisheries Service, which has jurisdiction over the salmon, are saying that they will need 6 years to complete this process. And that's an awfully long time. And so the parties are going to have to come to some understanding about either it's going to take 6 years or it's not, or if it does, what's going to happen in the interim. And the issue of interim remedies is a key one, because that may be where we are for the next 6 or 7 or 8 years.
That's very important in these intervening years. That sufficient surveys are done to get a good handle on how the Delta Smelt is changing, so as to maximize the information that's gained during that period.
I'd like to thank the panel for putting up with an ornithologist all day and asking naive questions. I'd like to thank CESAR, California Water Institute, Judge Manson, Congressman Costa, and Dr. Langley, and those of you who came. And we'll see you next time.
It doesn't stand out in the review paper and section. No, in the consensual. Yes, I didn't mean to be. So you're all typing up there or something. What? Chiefs are all split.
I did 3 of them. I did 3 out of 5.
No, on the other end.
Enjoy your trip to Yosemite.
Thank you for asking some astute
Oh, yeah.
questions.
Yeah, well, we got it. Your question was to make a damn decision, please. And the answer is but closer than they were 3 months ago.
Some part of it. Fresh eyes.
Are you here? No, I'm a bird guy. I'm in Minnesota. I don't think anybody here is
One question I was unclear if you
well, no, the panelists.
familiar with the hydraulic
I don't know.
system, and it's very, very interesting.
PhD in Berkeley. I had my PhD in Berkeley. It's out there.